Hello and welcome to the recording of the user guide for the GeneSetCluster R package. My name is David Ewing. I am the writer of this package, and if you're watching this you are interested in using it. So what we're going to show you is how to run the GeneSetCluster package, and I'm going to use the example script you can find on the GitHub wiki, and it's very simple. We start by loading the package. You can find this exact script on the GitHub wiki. So we start by requiring the package. Then we feed it in files of pathways. In this case, we are running an example of these are mice which have either wild-type genetics or a knockout for the TGF-beta receptor. And then we isolated microglia and macrophages, which we sorted by facts. And then we wanted to see, well, on a sequencing level, using DSEC2 pipeline, what are the differences between the two different cell types? And so this is microglia versus macrophages, and how are they affected by this TGF-beta knockout? We ran gene set analysis, pathway analysis using IPA, Ingenuity Pathway Analysis, and what we did is we exported those files. And so these are canonical files, these are untampered with, and what IPA gives you is a lot of data. So when we read, we need to require read Excel. When we read these files, this is what they look like. You have a pathway, you have a p-value, you have a ratio, which is how many of the expected genes are enriched, and they call it molecules or genes. It could be whatever, and those are then the main genes as evidence of why this is enriched. And as you can see, there's a lot of 364, but a lot of them are not significant. So it's a minus log p-value. So we want to filter this for significant genes, because that's what we want to feed into our analysis. If you're interested in non-significant ones, you could still use those, obviously. We want to load the wild type. We want to make sure that they're a data frame. We're going to require lemma because we're going to use the string split to function in order to count how many genes are enriched, because we would like our cutoff to be p-value 0.05 with at least 5 molecules. So that means we have to count the number of molecules. And to do that, we define a new vector, which we call molecule count. And then for 1 by 1, we just split it using the comma, because that's how they're split. And then we count how many are there. What's the length of mole? And then when we check it out, this is what it looks like. So we can see that in the first pathway, this TH2 pathway that we find, very, very significantly enriched. There's 17 molecules that cause it to be enriched. All right, then we do the same for the wild type gene set. And then we can filter, so we can create a significant version, which is 364 or 53 pathways, respectively. 53 or 281, respectively. And then we load this into our GeneSetCluster. And to do this, there is two ways of doing it. One is where all these steps are built into an automatic loading function, which is designed for either the GREAT pipeline or the IPA pipeline, because those are ones I personally run very, very often. But you can run any gene set analysis program. You basically need to feed it in what are the pathways. So in this case, what are the names of all the pathways? What are all the molecules? So to make sure that the program knows what are all the molecules. And then we have to say, well, this pathway belongs to this gene set, or this pathway belongs to this gene set experiment. You can run it with one experiment. You can run it with any number you want. So you can combine, in this case, two cell types in two gene sets, wild type versus scale, but you can add a knock-in, you can add other genes knocked out and compare. The idea of this package is that you can make it as wide as possible. So in this case, we just do a rep with the number of times we have pathways for every one of the groups. So our groups will look like that. Well, we got it from IPA, so that's a source. It's a canonical pathway, that's type. In this case, it's just metadata. But when you use the automatic loader, it wants to know whether it needs to load canonical pathways or not. In this case, structure means how are the genes presented, because sometimes they're presented as ensemble IDs or entrance IDs. In this case, they're gene symbols. and the program can automatically translate between them. They're separated by comma, as we can see, and we got them from mouse, so we feed it in the mouse org.mn package. All right, and now we have a GeneSetCluster object, which we called EPA wild type versus KO versus wild type. And then we want to combine them. And when we combine them, we by default combine them using the relative risk. There are two other built-in options. One of them is Cohen's kappa, which is this percentage of agreement, and the Accard index, which is the overlap between them. And depending on the number of genes you have enriched, one might be better than the other. RR works quite a little better for smaller gene sets. Accard works very well for larger gene sets. Cohen's kappa is somewhere in the middle, but it depends on what experiment you have. And then we want to determine, well, what's the number of optimal gene sets that we have. So there's this optimal gene set function. In this case, there's 3 different methods. There's the gap method, there's the elbow method, and the silhouette method. And then you have to feed it in, well, what's the maximum number of clusters that I can expect? How do I want to cluster? Do I want to have hierarchical clustering, or do I want to have k-means or something else entirely? You give it a title, and this takes a while, because it has to run a lot of iterations. In this case, gap uses bootstrapping. In this case, they say between 4 or 6, you can see a plateau. In this case, it advises 4 because the maximum increase of information is presented by 4, but there is more with 6. Play around with it iteratively. In this case, I did 6 last time. And when you run it, you combine. We combine using k-means. There is also hierarchical clustering that you can use. And then we have our cluster gene set. It's that simple. That's the many lines it takes to get there. And then you can plot that using networks. In this case, an RR min of 5 means, will there be a line draw between pathways? Yes or no, and when do you draw it? Well, you draw it when there's at least an RR of 5. If you use a Accard index, maybe these are usually between 0 and 1, so maybe you need to try 0.05. It all depends on your results. In this case, what we can see is that pink or reddish is in the center, surrounded by purple, surrounded by green, with a cluster of blue and a cluster of orange, and there's this little yellow box separate. If we try, well, what if there's a minimum of 10 relative risk between them? Then it's going to draw much less lines because the threshold is much higher. And therefore, the number of connections will be more limited. We can plot it in a heat map, so every pathway, with every pathway, and then the relative risk that we find will form the coloring. And as we can see here, it is separated by cluster and not by group. So if it's KO or wild type, doesn't matter, because cluster 1 clearly has elements of both, indicating there's a high overlap. Still, in this case, the overlap is not extremely high, but we are limited to the fact that the R goes really, really high. It goes to 120, and maybe we need to play around with the cutoff filter. We can also play around with the clustering of the order, what we want. So in this case, it's 1 to 6, but maybe we want to say, well, based on what we see, we think that cluster 2 and cluster 6 are more similar, because the way k-means separates things, when it's represented in a 2D space, it doesn't always necessarily line up. So in this case, let's say we want to do 1, 2, and then instead of 3, 4, 5, we want to do 6, and then we want to do 5, 4, 3. And we want to say, well, instead of 120, we want the highest color maximum to be 90. How does that look? In this case, it gives a warning, but it still works. And this is then what it looks like. So you can see that cluster pink and purple are very similar, and that's what we also saw in the networking pathway. All right, so now we know kind of what we want to show. How do we read this? How do we sort of get functional information out of this? Well, we can say we want an annotation of the molecules, and what that does is it's going to make it a little bit less legible in R. If you write it to PDF, it usually works a lot better. And you can play around with the size that you want. But basically you can see for every gene set, what are the genes enriched, and then you can see, oh, these are extremely the same, and they have a very high overlap, and the pathway. In this case, there's so many of them, you can't actually see it very easily. So when you write it to PDF, you can play around with the sizes and fonts, and it's much more legible. However, you can obviously play with the font size and make it much smaller. Instead of, you can make it 1 if you want. That's going to decrease the font size a lot, and then you can see a lot more on the plot. So in this case, you can see all the genes are there, but in this case they're so small we can't actually read them. So it's a bit of toggling around sometimes, a little bit. But that really depends on what you want to look at and where you're going to write it to, because in a PDF as a vector-based image you can obviously zoom in very much. Alright, so then let's say we want to see, well, how does it clustered, how does that look? Well, we can write it to a gene set with the right gene sets, and then our object, and then where we want to write it to. In this case, you can export both the distancing, the RR in this case, or you can just do the pathways and the clusters they are assigned to. You can also look at them by going inside of it using data, and then it's a list. And then we can see everything. Or to make it more easy, we can put it in the view. And this is what it looks like, our final data. So again, when we load things this way, there's not a standard p-value. We can assign it if we want, but it doesn't use it. It's more for selecting the pathways we want to take along. Molecule count the same, it's more for selecting, but the molecule count is actually very important for determining what kind of RR can we expect. So in cluster 1, the mean RR is 240, with the sum of it all is very high. You can use that to sort, because maybe if there's a higher mean, it's a more cohesive cluster. And sometimes what happens, I don't know if it will happen this time, but sometimes if you sort by name, you can see, oh look, there's the same name appearing twice in these two different very similar setups, but with one key difference. And this, you can see that the number of molecules in it is very different, and actually that the cluster it gets assigned to is not the same, indicating that the label is the same, but what's inside of it actually isn't. Well, if we want to then have a look at, well, what does this all mean? Like, can we use some tools to aid in our analysis? And the answer is yes. There are several tools built into the package that can aid in the analysis. One of them is string network. There's a string R package, which there's a plugin for. So then you can download, you can map every single cluster that you get. In this case, we ran for 6 clusters. We can run it through string, each of these 6 clusters, and you can do this for unique genes per cluster, or so the ones that make this cluster unique. Or you can do it for, well, no, this cluster has these genes, and so we just want to see what do these genes together represent. Alright, and we're back. So that took about 2 minutes to run, so I edited that out of the video, and basically what we get is we get a string object per cluster. So in this case we have 6 clusters, so then we can visualize 6 of them at the same time. And that will give you a string network for all the genes that are unique to that cluster. Which looks like that, and then if you just want to look at one of them, of course you can set the par to 1. And then maybe we're just interested in one of the clusters, because maybe that cluster has some of the most interesting things, and you can see, oh well, they're very closely together, they're very closely related. MMP14, CLC24, and those are the genes that are unique for cluster 5. And then there's another interpretation aid in the package, which is WebGestalt. WebGestalt offers over-representation analysis, and it's a very useful tool. It's online, it's free, but they have an R package as well, so there's a plugin to that. And what that does is it takes every cluster and it basically takes, well, these are the genes that make up this cluster, and then is there any kind of over-representation. Alright, and we're back, and this ran also for about 2 minutes, and then it gives you exactly 10, so the first 10 is the first cluster, and it tells you these are the functions, the Go terms, that are enriched in this cluster. And so in this case you can see they're very highly significant, using nominal p-value. Some of them are FDR significant, some of them are not. And so it's 60 in total because we wanted 10 per gene. And then what we can see is that, well, cluster 1 clearly has more lymphocyte migration, monocyte chemotaxis. The second one has more protein processing. The third one has regulation of complement factor, regulation this, regulation that. So it's more of a regulatory cluster. Third is a more metabolic cluster by the look of it. The fourth one is lymphocyte migration again, indicating that the fourth and first one are more similar. And so you can go through your clusters, you can go through your packages using these tools in order to help interpret what is the differences. And it works very nicely with multiple conditions. And it doesn't need to be EPA, it doesn't need to be great. You can use any tool you want. As long as there is a gene set name, there is a molecules enriched within, and you know what it is, then you can run it. Is it a symbol? Is it an entry ID? And that's it. And this is just a tool to aid you in your analysis. If you have any further questions, please contact me at ewoutewing@ki.se or leave a comment on the GitHub. Thank you very much.